In this video, we're covering mutations and their key role as a driving force in evolution. We're going to break it down by exploring the three major categories of mutations: point mutations, insertions and deletions, and chromosomal alterations. First, we have point mutations. Imagine your DNA as a long string of letters, each representing a specific instruction or word. A point mutation is like a typo in this string. A single nucleic acid, referred to as a coding sequence letter, is swapped out for another. Sometimes this swap changes the entire meaning of the word, and other times it doesn't. If the mutation does change the protein being made, it's called a non-synonymous mutation. If it doesn't, it's called synonymous. Think of it as swapping color C-O-L-O-R with color C-O-L-O-U-R. The meaning stays the same, but the spelling is different. That would be a synonymous point mutation. Next, we have insertions and deletions, often called indels. These mutations occur when multiple extra nucleic acids are added or removed from the DNA sequence. This can create serious disruptions, similar to shifting all the words in a sentence and making it unreadable. Imagine adding or deleting letters in a sentence. Suddenly, everything that follows doesn't make sense. These disruptions are known as frame shift mutations. They're particularly problematic because they can alter the entire protein, potentially leading to significant changes in an organism. Lastly, we have chromosomal alterations, which are changes that happen at a larger scale. These can involve swapping, duplicating, or even losing entire sections of chromosomes. For example, during a crossover event in meiosis, parts of chromosomes can be exchanged, creating new combinations of genes. This alteration is called a crossover event. They don't necessarily introduce new alleles in populations, but they do make it possible for new allele combinations to be passed on to future generations. Swapping alterations are called chromosomal translocations, where non-homologous chromosomes swap large portions of DNA in either a balanced or unbalanced way. Translocations are linked to conditions like cancer and infertility. In other cases, chromosomes don't separate properly, called non-disjunction events, leading to conditions like Down syndrome, where an extra copy of chromosome 21 is present. These alterations can lead to significant evolutionary changes by introducing new gene combinations or disrupting normal development. So, to sum up, mutations, whether they're point mutations, indels, or chromosomal alterations, are vital to evolution. They create the genetic diversity that allows species to adapt and thrive in changing environments. Understanding these mutations helps us appreciate the complexity of life and the ongoing process of evolution.